Hello everyone, welcome to my channel. Before starting the video, please subscribe to my channel for more such videos. Today's video is on fatty acid synthesis. I have made another video on fatty acid oxidation. Link has been provided in this description. Fatty acid synthesis is a chapter in metabolism of lipids. Now, why there is fatty acid synthesis? The dietary carbohydrates and amino acids, when consumed in excess, they are converted to fatty acid and stored as triglycerols, so that when there is need of energy in case of starvation, they can be used. For fatty acid synthesis, the source of carbon atom is from acetyl-CoA, and acetyl-CoA is known as building block of fatty acid. Reducing equivalents NADPH, and energy supplied for FA synthesis is by ATP. Now, where is this fatty acid synthesis occurring? The enzyme machinery is in cytosol, and de novo or new fatty acid synthesis occurs in liver, kidney, adipose tissue and lactating mammary gland. Now there are stages. Just like beta oxidation, there are three stages of fatty acid synthesis. First, production of acetyl-CoA and NADPH. Second, conversion of acetyl-CoA to malonyl-CoA. Third, reaction of fatty acid synthase complex. Let's see the stage 1, that is production of acetyl-CoA and NADPH, as well as the transfer of acetyl-CoA from the mitochondria to cytosol. So the pyruvate produced in cytosol or cytoplasm is transferred into the mitochondria and with the help of PDH is converted to acetyl-CoA. The acetyl-CoA formed is converted to citrate, and this citrate goes into the cytosol from the mitochondria. Then the citrate in cytosol divides into oxaloacetate or acetyl-CoA. Acetyl-CoA formed in cytosol from citrate with the help of NADPH+, which is formed by HMP pathway, goes into the lipid biosynthesis. Step 2 or the stage 2 is formation of malonyl-CoA from acetyl-CoA. Now the acetyl-CoA that is in cytosol, it undergoes the activity of acetyl-CoA carboxylase in presence of biotin, and it requires ATP and CO2, and it forms malonyl-CoA. Now this malonyl-CoA goes into the stage 3 where there are reactions of fatty acid synthase complex. So there's acetyl-CoA, acetyl ACP and malonyl ACP together acting with the help of enzyme beta-ketoacyl ACP synthase to form acetoacetyl ACP. This acetoacetyl ACP undergoes the reaction to form beta-hydroxyacyl ACP in presence of beta-ketoacyl ACP reductase. This beta-hydroxyacyl ACP then forms trans-enoyl ACP by enzyme 3-hydroxy-ACP dehydrase. Trans-enoyl ACP then goes and forms butyryl ACP, 4-carbon ACP. So that is by enoyl ACP reductase. Then there is addition of malonyl ACP and their formation of hexanoyl ACP, that is 6-carbon. Then again there is addition of malonyl ACP and the cycle repeats till we get palmitoyl ACP, that is 16-carbon ACP. And then there is addition of water by which ACP is removed and palmitate is obtained. Now there are two places where NADPH plus H plus is used. First is the beta-ketoacyl ACP reductase stage, where there is conversion from acetoacetyl ACP to beta hydroxyacyl ACP. Second stage is trans-enol ACP to butyryl ACP. So you can say that each malonyl ACP molecule requires 2 NADPH plus H plus. Now this FAS complex, fatty acid synthase complex, it's actually a dimer, and each monomer has 7 different enzymes and acyl carrier protein, that is your ACP. Now palmitate synthesis, there are 16 carbons in palmitate. So 2 carbon come from the acetyl-CoA directly and rest are from malonyl-CoA. Now you can see here a reaction. 8 acetyl-CoA plus 7 ATP plus 14 NADPH plus 14 H plus giving palmitate 8 CoA, 7 ADP, 7 inorganic phosphate and 6 water molecules. Now how is this reaction forming? Now we can say why 8 acetyl-CoA, because there are 16 carbon in palmitate, so you require 8 acetyl-CoA. Now out of these 8 acetyl-CoA, 7 are converted to malonyl-CoA, and hence in the conversion of acetyl-CoA to malonyl-CoA you need ATP, so there are 7 ATP. Then each malonyl-CoA requires 2 NADPH plus H plus in the reaction of FAS complex. So there are 14 NADPH plus H plus 14 H plus for 7 malonyl-CoA, and this is giving you palmitate. Then we are seeing regulation of fatty acid synthesis. Acetyl-CoA carboxylase. It is a rate limiting step for fatty acid synthesis. This enzyme is for the conversion of acetyl-CoA to malonyl-CoA. Citrate increases the activity of acetyl-CoA carboxylase and hence it stimulates fatty acid synthesis. Palmitoyl-CoA and malonyl-CoA inhibits fatty acid synthesis. Then there is hormonal regulation. Enzyme is active when it is dephosphorylated. CAMP-dependent phosphorylation hence inactivate the enzyme. Glucagon, norepinephrine and epinephrine, they promote CAMP-dependent phosphorylation and hence inhibit the fatty acid synthesis, whereas insulin promote fatty acid synthesis. The next regulation is dietary regulation. If your diet has more carbohydrates and it is fat free, or there is sucrose present, then it promotes fatty acid synthesis. But if your diet is not there, or you can say when you're starving, or if you have fat diet, then there is fatty acid synthesis decreases. Then NADPH availability. NADPH is obtained from HMP shunt pathway or from citrate. So this NADPH is required in the stage 3, that is reactions of FAS complex. So this availability is important. Then there is serum free fatty acids. These are inversely proportional to hepatic free fatty acids. So this is simple regulation of fatty acid synthesis. Thank you. We'll meet in the next lecture